Nucleus, the brain of a cell. History. First discovered and named by Robert Brown in 1833. Term Nucleolus, Bowman in 1840. Term chromatin, W. Fleming in 1879. Occurrence and position. Occurrence found in all eukaryotic cells of plants and animals, exceptions mature sieve tube and erythrocytes. Prokaryotes lack true nucleus. Position usually at center but change from time to time according to metabolic state. Staining properties. Fluorescent dyes with aromatic amino or guanidine groups such as propydium iodide, ethidium bromide, diaminophenyl indole, acridine orange and Hoech's dyes interact with nucleotides to emit fluorescence. Morphology. Number. Mononucleate cells, binucleate cells and polynucleate cells. In animals we can see syncytial cells, in plants cenocyte. Shape of a cell. Shapes: spheroid, cuboid, isodiametric, ellipsoid. Example: squamous epithelium, discoidal in shape. Leukocytes, it's irregular in shape. Size: generally occupies 10% of cell volume. Size varies from 3 μm to 25 μm depending on cell type. Size of nucleus is directly proportional to cytoplasm. R. Hertwig gives formula for determination of size. NP is equal to Vn by Vc minus Vn, where NP is nucleoplasmic ratio, Vc volume of cell, Vn volume of nucleus. Size is related with ploidy level. Example: haploid cells have small size nucleus than diploid and polyploid cells and vice versa. Thus, size depends on cell volume, amount of DNA and protein, metabolic phase of a cell. Now we can see the diagrammatic representation of a nucleus or the structure of a nucleus. Ultrastructure. Nuclear envelope encloses DNA formed from spherical inner nuclear membrane, contains special proteins, act as binding site for IFs, nuclear lamina, outer nuclear membrane, continues with ER, started with ribosomes, proteins, perinuclear space 10 to 50 nanometer diameter, contains fibers, lipid droplets, crystalline deposits. Nuclear lamina, protein meshwork, 50 to 18 nanometer thick, lines inside surface of nuclear inner membrane, plays crucial role in assembly of interface nuclei. Here we see the nuclear envelope in a diagram. Nuclear pores. Structure: circular in shape, diameter 10 nanometer to 100 nanometer. Nuclear pore complex have octagonal symmetry, contain 2 rings, large particle that form central plug and radial spokes. Proteins: integral membrane protein, a glycoprotein of 120 kDaltons, transporter protein 63 kDaltons. Now we can see the pore density correlated with transcriptional activity of cell, RBCs and lymphocytes. 3 pores. Proliferating cells in liver, 15 to 20 pores. This is the diagram of nuclear pore complex. Transport through nuclear pores. If cell is synthesizing DNA, it needs to import about 10/6 histone molecules from cytoplasm every 3 minutes for packaging DNA. mRNA complex with special proteins forming ribonucleoprotein particles are actively exported through nucleus. Movement of granular material through a nuclear pore is shown as a diagram. Now we can see the transfer of proteins through a nuclear pore. Next we are going to see the nucleoplasm. Transparent, semi-solid, granular, slightly acidophilic, also called nuclear sap, nucleoplasm and karyolymph. Composed of nucleic acids, deoxyribose nucleic acid, DNA, ribose nucleic acid, RNA. Proteins. Complex proteins categorized into basic proteins take basic stains, so the name is given. Example: nucleoprotamines or Histones. Non-histones are acidic proteins. Example, phosphoproteins. Enzymes. Contain enzymes necessary for DNA RNA synthesis. Example, polymerases, NAD synthetases, adenosine deaminases, etc. Also contains cofactors and coenzymes like ATP and acetyl-CoA. Lipids. Small lipid content. Minerals. Contain several inorganic compounds such as potassium, calcium, sodium, magnesium, etc. Chromatin fibers. Thread-like coiled and much elongated structures take basic stains like basic fusion. Observed only during interphase. Chemically made of DNA and proteins. Protein. DNA 1:1. Types. Heterochromatin, darkly stained condensed region, supposed to be metabolically and genetically inactive because it contains small amount of DNA and large amount of RNA. Euchromatin, lightly stained, diffuse region, genetically active, large amount of DNA present. Now we can see the spaghetti model for organization of chromatin in the interface of a cell nucleus. Next we can see nucleolus. Prominent spherical, colloidal, acidophilic body, bacteria and yeast lack nucleolus. Size depends on synthetic activity of cell. So sperm cells, blastomeres have smaller nucleolus than oocytes, neurons, secretory cells. Number 1, 2 or 4 depending on number of chromosomes. Position. Eccentric. Associated with nucleolar organization represents 2-degree constriction of NO chromosomes. Chemical composition. Not bounded by any limiting membrane. Contains NO DNA, 4 types of rRNAs, 70 types of ribosomal proteins, RNA binding proteins and RNA splicing nucleoproteins. Now we can see the ultrastructure of a nucleolus as seen through a microscope. The nucleolus contains phospholipids, orthophosphates, calcium ions, also contains enzymes like acid phosphatases, nucleoside phosphorylase, RNA methylases. Ultrastructure and function. They are sites for biogenesis of ribosomal subunits. Here 3 types of rRNAs are transcribed from precursor 45S transcript which undergoes processing with the help of protein. Structure of nucleolus. Fibrillar center represents innermost region of nucleolus. RNA genes of NO of chromosomes are located here. Transcription of these genes initiated here. Dense fibrillar component. This region surrounds fibrillar center. RNA synthesis proceeds where 70 ribosomal proteins binds to transcript in this region. Cortical granular components, outermost region, processing and maturation of pre-ribosomal particles. Mitotic cycle of nucleolus. Appearance changes during cell cycle. During mitosis and meiosis, nucleolus disappears during prophase. As cell approaches mitosis, RNA synthesis stops. Nucleolus first decreases in size and then disappears as chromosomes condenses. In telophase, RNA synthesis restarts where tiny nucleoli reappear at the chromosomal locations. Now we can see the general functions of a nucleus. Functions of nucleolar genes are to produce the ribosomal RNA molecules which forms ribosomes. It has a vital role in protein synthesis. Nucleolus of nucleus plays vital role in cell division. If nucleolus gets injured, cell division stops. Chromatin plays an important role in the inheritance of characters from one generation to another. Nuclear sap is the site of enzyme activity. It is supposed to participate in formation of spindles during cell division. The nuclear pores regulate exchange of materials between nucleus and cytoplasm. As in the whole, we can see nucleus is a part of a cell which is very important to the life of a cell. Thank you.